Hello, my name is Malea Nguyen, and I am a fourth-year biological sciences student concentrating in biotechnology. Over the summer, I conducted research in Dr. Teskey's lab at the Hotchkiss Brain Institute of the University of Calgary. Today, I'll be presenting all my findings from the summer, which is about how hyperoxia after febrile seizures led to lipid peroxidation within the hippocampus of neonatal rats. As a short introduction, febrile seizures are seizures that are associated with fevers. They are common among children between 0.5 and 5 years of age. In neonatal rat models, febrile seizures result in an increased amount of oxygen in the brain for a prolonged period of time, which is called hyperoxia. As shown by this figure on the right, hyperoxia leads to the formation of reactive oxygen species, which can cause damage to cells, proteins, and lipids. This forces the cell into a state of oxidative stress that is time and concentration dependent. The brain is especially at risk for oxidative stress as it requires a disproportionate amount of oxygen for its size, since the brain is constantly needing to metabolize and use energy. As demonstrated by this figure, reactive oxygen species can lead to lipid peroxidation, which is also particularly inclined in the brain due to the high fat content of glial cells and myelin. Due to oxidative stress, neonatal hyperoxia can lead to difficulties in memory recognition in adulthood. This brings me to the objectives of my research, in which the study will examine hyperoxia inhibiting drugs and its efficacy in mediating oxygen in the hippocampus and reducing oxidative stress. The study will test whether the drugs are as effective when provided after a febrile seizure, which will be my post group, compared to before the febrile seizure, which is my pre group. In this experiment, I used lipopolysaccharide or LPS to cause general inflammation in the pups, as this increases the expression of anandamide or AEA in this figure. AMG 9810 will be studied as the first drug, and it is a transient receptor potential vanilloid 1 or a TRPV1 inhibitor. TRPV1 is a receptor for anandamide, which causes a vasodilation response that may lead to neuroinflammation. The study examined 4 groups: AMG 9810 pre, so my treatment group, seizure post, no seizure pre, and my no seizure post group. At postnatal day 8, or P8, an electrode and optode are surgically implanted into the dorsal hippocampus to measure the EEG and the oxygen levels, respectively. On P9, the treatment and seizure groups were given an IP injection with LPS at a dose of 0.2 milligrams per kilograms. The no seizure groups were given 0.1 mils of 0.9% saline instead of LPS. The doses doubled for the following 2 days to 0.4 milligrams per kilograms for LPS and 0.2 mils for saline. This is the P12 timeline, which is the febrile seizure day. This timeline outlines the appropriate dosage times for all 4 groups. Essentially, the pre-groups were given their drug prior to being exposed to endogenous heat, whereas the post-groups were given their treatment after being exposed to heat. The drug for the no seizure group is simply just saline, and the no seizure groups were also exposed to heat for only 20 minutes before being removed from heat, whereas the treatment and the seizure groups were heated until a seizure was induced. After removal from heat, all 4 groups received a sub-Q injection of 0.4 mils of 0.9% saline. After sacrifice, the brains were removed and then stained using immunohistochemistry of 4-HNE, which is a lipid peroxidation byproduct of oxidative stress. These preliminary results show that the O2 levels were not significantly different from the treatment in the no seizure groups. The results also show that O2 levels in the seizure group were higher in both the treatment and the no seizure groups for 50 minutes onwards. These results here also show that lipid peroxidation can be seen in the hippocampus for both the seizure and the treatment group, as visualized by this green 4-HNE. The lipid peroxidation can especially be seen in the dentate gyrus, which is this nose pin area in the middle of the scans, as visualized using DAPI. So, in conclusion, the dentate gyrus in the hippocampus are continuously restructuring throughout life during the learning and reconsolidating processes, and thus are at further risk of dysfunction. The next steps may be to examine whether dentate gyrus dysfunction due to lipid peroxidation could be the link between neonatal hyperoxia and a decline in memory recognition. Further drugs will be tested, such as a CB1 inhibitor, as well as my AMG 9810 post and a seizure pre-group. A separate cohort will be examined in January using a novel object recognition test to assess disruptions in memory recognition at a later age. These are my references for this presentation, and that concludes my presentation. So thank you very much for listening.